very close.
Yeah. The second speaker is going to be Professor Philippe Dumas. Philippe is a research director of Biophysics and Structural Biological Group at the French National Center for Scientific Research in Strasbourg, and their research interest is in the biophysical and structural aspect of biology, as well as in quantitative modeling of regulation mechanisms. They put a strong emphasis on using ITC as a transversal experimental method. In the talk, Phyllis is going to stress the kinetic ITC shed light on RNA-mediated genetic regulations.
Yeah, thank you. Okay. Hello, everybody in the room, and not only in the room, because I understand that there are people all over the world hearing us. So hello, everybody. I am Philippe Dumas, and our team is doing biophysics. We are deeply involved, historically at least, and also we're doing crystallography and structural studies, but we are more and more involved in biophysics studies in an institute in Strasbourg. And the name of our unit is ARN, RNA, ribonucleic acid in English, okay? And so we are deeply involved in studying RNA. So there are other macromolecules and proteins, as was mentioned before. So RNA is one of them, and so we were very interested in using ITC, and particularly we have developed a new technique, so kinetic ITC. And as written here, you see, it sheds a lot of light on RNA-mediated regulation. Okay, so I will divide this talk in two parts. One is, let's say, technical, because this is the right place for doing that, okay? It's a technical part on what is kinetic ITC. And the second part, I will go in the biological part and the application to show you how it helped us a lot understanding the kinetic mechanism of kinetic regulation with RNA. So we have published this technique some time ago, not 2 years ago, in JACS. This is the paper, and there is a website indicated in the paper, so that allows you to have some demonstration. I will show you a small of them in a minute. Okay, and I would say that it turned out that this kinetic study, to be fair, the fact that ITC could be used for doing kinetic studies, which is not new. You know, there are a lot of time that kinetic studies have been performed with ITC, particularly with enzymatic reactions, so it is not new. But at the same time, so people also using RNA, folding of RNA, has used also the possibilities of obtaining kinetic information by ITC. But I think our technique that we have developed is more general. So just for those, I think most of you, if not all of you, are aware of the basics of the ITC. I want to have two slides to show clearly what we are speaking about. So we have a compound A that is in the sample cell, and we inject the compound B, okay? And we look at the reaction taking place, and during the reaction, when you inject B, then you have, for example, in the most simple case, formation of a complex, and you have either heat production, it is exothermic, or heat absorption, it is endothermic. And the very important fact that has to be recalled here, the output is not a heat energy, it is a heat power, microcal per second or microjoule per second. And this is why we can do kinetic studies, because this heat power is directly related to the kinetic of the reaction. So in fact, here you have an example of experiment that we have done. You see E. coli Ti-C riboswitch. This is what I will explain you later. So the very nice experiment, when you look at them at the different injections, you see very well, it appears that there is a significant variation of equilibration time from injection to injection. You see that the last injection, it takes more time to go to the baseline in comparison with the first injection. And in fact, all of those practicing ITC have observed situation of quick return to baseline and situation of slow return to baseline. So to emphasize that the fact that we have kinetic effect in ITC, this is an example of what we have obtained. It was also on this riboswitch at different temperatures here. So the same riboswitch at 3 different temperatures. And you see very well that at 35 degrees, the things are going faster than at 15 degrees. In fact, at 15 degrees, we were a little bit beginners at that time, so you see that we did not leave enough time for the injection to finish, and everything goes more slowly. So this is clearly an indication of a temperature effect on the kinetic of a reaction. And here's another example that, if I remember well, Eric in IFA was in the audience. It is reverse transcriptase, isn't it? Yes? He will mention that in his talk later. So you see that in that case, you have a very slow kinetics of return to equilibrium. It takes at least 10 minutes to go back to equilibrium. So clearly, what we see is that there is kinetic information in this titration curves, not titration curves, in the injection curves. So the question is how retrieving this kinetic information and how transforming ITC into kinetic ITC to obtain both thermodynamic and kinetic information. So in some way, and as you will see, what we get with ITC instrument in the good situations, we get at least as much information as with SPR. So now I go back. I go to the real little technical part of the talk to explain you. So let us suppose you have simple situation. A plus B gives a complex C. So you have, of course, a delta H of formation of the complex. So let us make a very basic kinetic chemistry. This is a second order kinetic reaction. And so you can consider what is the variation of the concentration of C. You have what comes from the left, the terms Kaon, and you have minus, which goes back to the left, so minus Kf, the concentration of C. This is very basic thing. So, and it turned out that the link with what is measured, because we obtained by this kinetic equation of chemistry, the derivative of the concentration of C, it is immediately linked to the heat power that is measured, because it is the product of the delta H of the reaction in calorie per mole times the volume of the cell multiplied by the derivative of C. Indeed, calorie per mole multiplied by mole per second, you obtain the term on the left, calorie per second. And in fact, this very basic equation, very basic link, so it is all what is necessary for you to understand the basis of key in ITC. Of course, provided that one skips a few technical details, so that not secondary, and I will say a few words about them. So no, before going to these technical details, a very, very important point for the success of Keen ITC. So in fact, ITC, because what we measure in ITC, as all of you know, we measure the delta H of the reaction, okay? And this is extremely important because there is a very important and rigorous equation in thermochemistry, which is the Van 't Hoff equation, which relates the variation of the equilibrium constant versus the temperature to the delta H of the reaction. This is not an approximate reaction equation. This is a rigorous equation. Everything that you can do in practice to analyze data, if they violate Van 't Hoff equation, something is wrong. So it is extremely important because we measure the delta H. This is exactly what we measure. Therefore, what we know is the variation of delta H with the temperature, and therefore, with this, because this is measured, we can integrate the equation, and therefore we obtain the variation of the equilibrium constant with the temperature. And so that means that only one value at one temperature is free values. Then, provided you know the equilibrium constant at one temperature, you should know it at all other temperatures by the integration of the Van 't Hoff equation. So why is it so important? Because, of course, you have an obvious link between the equilibrium constant and the kinetic parameters, and therefore, if you have K on and K off, the two kinetic parameters, only one of them is unknown, because if you know K on, you know K off, because it is related by this to the equilibrium constant. So the conclusion is that suppose the K on, for example, or K off, let us choose K on. Suppose it follows the Arrhenius equation, which is a good way of representing the variation of a kinetic parameter of the temperature, then, provided if you know these two parameters, K on at some temperature and delta H, the activation energy, then you can compute the evolution of K on at all temperature. Therefore, with two parameters only, K on 0 and activation energy, you can represent the K on at all temperatures, and because you know the Ka, the equilibrium constant at all temperatures, that means that you know also the K off at all temperatures. So I hope you have understood the extreme importance of the Van 't Hoff equation in this matter to make a strong link between thermodynamics and kinetics. So now the practical problems, because they are real practical problems. The first one is that the injection of compound B is not instantaneous. So when you want to make kinetics, as you will see, it is extremely important to take that into account. The mixing also of compound B is not instantaneous. And very important, the instrument does not respond instantaneously. There is a response time, which is in the instrument that we are using, the Malvern instrument now, it is of the order of 3.5 seconds. So let us, I want to illustrate this with some animation to show you the different effects. So, yeah, help me. Thank you, Andrel. Yeah, okay. So here, where is this? No. Here? Okay. So here you have an injection. Okay, what we can do, a particular injection of an experiment. We can, for example, superimpose all the the experimental injections, okay? So let us not, I don't focus on that now. And so you can look, of course, so it's not very easy to move that. Yes. You can, of course, look at the effect of the delta H. Of course, if you change the delta H of the reaction, it will obviously, it's not very, okay. Okay, so now I want to apart these obvious things, let us look at the effect of the k-on of the reaction, this parameter here. If we change it, if you increase the k-on, of course you see that the reaction goes faster, and of course it changes. So of course it is. So why we can do kinetic ITC? Because you see the k-on of the reaction makes change the blue curve into the red one. Importantly, the integral between the two curves of the two curves are the same. The heat of the reaction is not changed, but the kinetic of the reaction is changed. So this is the first thing. So of course we can look at the influence of this, the response time of the instrument. So if you change the response time of the instrument, you see that it will influence as well the kinetic of the how the injection curve appears. Slow. And I will yes, maybe. I will show you the effect also. You see that the mixing time, here the mixing time, it is 0.8 seconds. If you change it a bit, you see that you displace as well, you change. So all these parameters are important for describing exactly each injection curve, and therefore in the software we have developed, so everything is taken into account and everything is corrected so that we can accurately describe each injection curve. So, okay, let us go back to, oh yeah, thank you, Angel. Okay, so now, okay. So now, okay, quickly. Can you go because there are many? I'm sorry. You can, yeah, good. No, yeah, okay. Okay, now I want to come to the biological part and to show you how using KINE-ITC allows us to understand a complex RNA-mediated regulation mechanism. So I need to introduce a little bit the part, okay? What is our subject? Subject is we are interested in the biosynthesis of thiamine pyrophosphate, TPP in short, which is represented here on the right. So the one part of the molecule you see which is boxing is in vitamin B1 that you need to get because you do not produce it, and the whole molecule with a phosphate on the right, so the pyrophosphate is thiamine pyrophosphate. And this is what is important. It is a very important and universal cofactor for many enzymes that was written on the left. And in particular, it is a universal cofactor in the pyruvate dehydrogenase complex, which is a Krebs cycle. So for this reason, the level of TPP has to be tightly regulated. And as you see now, the biosynthetic pathway of this TPP is complex. So here, sorry, the slide is not very nice, but you have, we don't need to go into the details. What you have at the bottom in the green box is thiamine pyrophosphate, which is the end product of the biosynthetic pathway, and you have many genes interacting to build up this complex molecule. This is in bacteria, that. And, okay. Yes, and so you have a negative feedback. When there are enough TPP, then you have negative feedback on the messenger RNA themselves through riboswitch. So what is a riboswitch? So probably some of you are not aware of this. A riboswitch is a structured part in the messenger RNA, so in bacteria, in the 5' untranslated part of the mRNA. So you see here that you have the AUG codon here. So that means the mRNA goes on the right, and here you have, for this gen M in E. coli, so here you have the structured part of the non-translated part of the mRNA, which can bind here the ligand, the TPP, or not. And depending on whether or not the ligand is bound, you sequester this sequence, which is the Shine-Delgado sequence, which interacts with the ribosome, okay? And if the ligand is not bound, so it is free, the ribosome can bind, or if the TPP is bound, then this sequence is occluded and the ribosome cannot bound. Therefore, you have a translational regulation because sequestration of this Shannon-Dalgarno sequence. So this kind of riboswitch can regulate translation. But this exactly the same kind of riboswitch, very related, in another messenger, here in the TIC, which are also in the big pathway. So they can regulate by the same kind of thing, transcription regulation. They can regulate transcription because there is, if you have the TPP, you have formation of this hairpin here, which makes the RNA polymerase, which will detach prematurely the RNA polymerase, and so the messenger RNA will not be transcribed. So you can have with very alike structures, you can have either translation regulation or transcription regulation. Okay, so here is just to show you what it means in 3 dimensions. This is the crystal structure for this related riboswitch. So you see very well here in 2D here the binding of the ligand here, and here in the crystal structure how it is. And you can think of this ligand as a piece of string which holds together the two halves of the molecule you see. And so the binding of the ligand stabilizes the riboswitch. Okay. So what, in fact, this is what I'm telling you today is the result of a long work. In fact, we were interested in the kinetics of folding of this riboswitching upon TPP binding. We were interested in the thermodynamic and kinetic, of course. And we wanted to have a biophysical view of the regulation mechanism. This is what I will explain. So we have used different techniques, and particularly KINE-ITC. But today, I will focus essentially on this, okay, on this thermodynamic parameters and the regulation, the mechanism itself. So we want to regulate gene expression, okay? So what you have here is the curve, the probability of the riboswitch to be in the on state. That means which lets produce the TPP. But so we have, the question is where should we regulate, around which concentration? So I make a long story short. In fact, by data mining, we came to the conclusion that, I go fast, TPP regulation should occur very roughly around 3 micromolar. When the TPP concentration is below, let's say, micromolar range, then the bacteria should build up TPP, but when it is significantly above 3 micromolar, there is no need of building up, of synthesizing the TPP. So here the regulation curve that we should have. So because this is, as you will see, this is the kinetic part is very important in this thing. So we have to answer the question, did we get a single binding step with a single kinetic step with K on, K off, or did we get 2 kinetic steps with, for example, in that case, binding first, the first binding step here with a K on and a K off, and then here, so it is induced fit, and then a change of conformation with 2 parameters, K folding and K unfolding for the RNA, or do we have 2 kinetic steps, but with a binding in the second part here, in the change of conformation before? So I make a long story short again, and in fact, so we show that a single kinetic step cannot explain our results, and that this was the correct kinetic mechanism. So it is an induced fit, binding first, and change of conformation later. So here, to summarize that, I show this picture. So here you have the riboswitch, which is represented here in the crystal structure has been opened to represent a somewhat unfolded structure. And you see here the TPP arriving here, and it binds. So you have a K on and a K off for this binding step, which gives you B1 for the bound state number 1. And then you have a change of conformation, and you have the state B2 for bound state number 2. So it is extremely important here for the understanding of the following, to make the difference between the different KD. Here you have the KD corresponding to this initial binding step equal to K off over K on. But it is different to the overall KD of the overall reaction, okay, which relates this concentration to these 2 concentrations. I name it KD overall, and they are not equal. So, keep in mind this difference between the KD of this initial binding step and the KD overall. So now, what we, okay, because we have two steps, of course we have the K on K off here, and we wanted to get it. Because we have a second step, we wanted to get these parameters KF and KU, but because we say that we have a initial binding step, that means that here's a delta H of binding during this initial step, and there is a delta H of folding for the second step. And we want these two terms, and of course the sum of these two terms, delta H of binding plus folding, has to be equal to what you measure on the calorimeter. And also, the equilibrium constant that is determined by ITC has to be equal to the product of the equilibrium constant here and here, the product of these two equilibrium constants. So now, I show you the results. Okay, we have applied this KIN ITC, and here you see the fit of experimental data for one of these Ribot switches at different temperature. Here you see it is very important to perform experiment at different temperature. And so you see here, what you have here is a superimposition of the experimental points. They are not clearly visible here, and you have the theoretical curve. So you see that the fit is quite decent. The fit is, so all the injection curve of all experiment have been fitted, and they are fit all together, okay? No, not fitted separately, which is very important. This is the global fit of everything. So now, let us have a look to understand that. So the interest of these results. So the result of the KIN ITC, as a result, you obtain function of the temperature. You do not obtain discrete values, you obtain function for everything, and you can plot them. So here, for example, you have the evolution of the KD overall, you remember, the overall KD as a function of the temperature, and you have here the evolution of the different delta H term. I will not focus on them today. And you have here the evolution of the KD of the initial binding step. You remember the two. So we will focus now on these two. These two things, this one and that one. And let's have a look. You see that the KD overall is of the order of 0.05 micromolar, which is clearly too small value for regulating, because if the regulation were thermodynamic, classical regulation, that means that at around 3 micromolar, the Ribot switch would be always saturated by its ligand. Therefore, we can conclude that we cannot have a classical regulation by mass action law. We have to have a kinetic regulation, and this is what I will explain later. And what is interesting is that if you look at the KD of the initial binding step, it is right in the right range for regulating around 3 micromolar. You see that the value here is exactly around 3 micromolar, which is exactly what we want to get. So what would it mean if we had a KD overall, if the regulation were classical one, around the KD overall? We would have regulation around this point, around 0.05 micromolar. But in fact, we want to regulate, as I tell you before, we want to regulate around this concentration. And clearly, the variation of the probability of the Ribot switch to be in the on state is too small. You cannot take a decision with such a small. So that means that we would like to have, for a classical regulation, we would like to have another KD overall around, for example, 1.5 micromolar. Then it would be very nice to regulate around that concentration. But we don't choose. The overall KD is not around 1.5. So how can we do? So KD overall is not adapted. How to solve this problem? No? So as I tell you, is kinetic regulation. What does it mean exactly? How does it work exactly? I want now to go into a very simple comparison, but I claim this comparison is an exact one. It's exact comparison. Okay, why? Consider different springs and masses, okay, in this Physics Institute, it is the right place for speaking of that. And so this is equilibrium measurement. You have different masses, suspended to the same spring, so we have different length. This is equilibrium experiment. The weight is equilibrated by the tension of the spring. Perfect equilibrium experiment. And you see that you have different measurements. That means that you can take a decision. You can decide, for example, to consider, you can decide to take all spring above this line, for example, and you have different, you can select them on the weight, on this equilibrium measurement. Okay, now consider a situation where the masses are too heavy. The springs are completely elongated, they cannot. So what does it mean? You are at equilibrium, but as you can see here, all of them give you approximately the same reading. So you cannot take any decision based on the equilibrium measurement. So let us make another kind of measurement. Let us make kinetic. Let us make a rate. For example, we can ask when will each mass pass at some point during the fall. For example, when will it pass here? And you let them, oh yes, you let them fall. Normally it should be a continuous fall. And you see that they pass at this line at different times. So there is a race and they pass at different times, although at the end they all reach the same position almost. So if you put a vertical length apart, you see here you can let all them pass, but if you displace this vertical line in time, then you will select different masses, so you can take a decision based on this race. If you still displace the line, of course, you will select different things. Okay, so here you have a very good illustration of what is the kinetic measurement. And so let us say in few words what it means. A balance can select the correct weight from time measurements, even though it would not distinguish the different weights at equilibrium. Okay, so the question is, how could a riboswitch select the correct ligand concentration, even though all concentration would saturate almost equally the equilibrium? So in fact, it turned out there is an answer to that question. You can ask the RNA polymerase, and this is now a very well-studied subject, you can ask the RNA polymerase to make a pause before riboswitch synthesis. This has been described several years ago for the riboswitches. And so the rationale is of measuring the ligand concentration before equilibrium has been reached. And if the concentration is not high enough, the ligand has not enough time to bind. Okay, so what does it mean? You see here you have the RNA polymerase, which is synthesizing the riboswitch. And here, in fact, you have a pausing step. The RNA polymerase can make a pause, and so the ligand can bind or not. And during this pausing time, you can make the kinetic experiment. Okay, so again, here I go fast in the story because we have developed a kinetic model of this regulation, and we obtain this function for the probability of the riboswitch to be in the on state are the function of the concentration of the ligand here. So this function is a part of is a sum of several terms. Here, this part corresponds to the blue curve. It corresponds to what would happen if the riboswitch was regulated at equilibrium. And now comes the second kinetic part here, which corresponds to the bell-shaped curve here. This sum of two exponential. What is important here? Thank you, Angel. And so the sum of the two curves is the regulation curve, the overall kinetic regulation. The important point is that with the value that we have obtained by KINA-ITC, we can compute all parameters which are here, F2, S2, K fast, K slow. We can compute all of them with the parameter that has been obtained by KINA-ITC. That means we do not have any free parameters in this equation, okay? And so now we can have a look to the, very important point. In the model, you have this time Ts, which is a pausing time of the RNA polymerase. So if you, now I come to an interesting result of this analysis. we have seen that this is a purely mathematical result. We've seen mathematically that if the posing time of the RNA polymerase, if it is related to the reciprocal of k off, here, this k off, okay, then the regulation is exactly around the KD. You remember this KD, which is ideally suited for the regulation, around 3 micromolar. Then if we have this link of the RNA polymerase posing time, if it is related to the reciprocal of k off like this, then we will regulate exactly around the correct KD, not around the KD overall, around the good KD. Okay, so now we can ask the question, does it make sense? This is a mathematical result. Does it make sense? So here I show you curves which have been calculated with all our results for the 2 different riboswitches, T tie C tie M, and you have the regulation curve in colored, okay, with different times, different RNA polymerase posing times. So you see that when you change the posing times, you displace the regulation. In terms, you regulate around different concentration. And here, the medium curve here, it has been calculated with this Ts equal the reciprocal of k off. And it turns out so that first, so the time that we obtained, so you see here 11 seconds and 24 seconds, the medium curve, it turned out that these values are exactly of the order of magnitude of what is observed for the RNA polymerase posing times in real situations. Therefore, our K90C result falls well with the observed value of RNA polymerase posing time. And the second good point is that when you look at where it makes the regulation at the inflection point here, in both cases, so you see that it falls around the correct concentration. It regulates around in the micromolar range as it should be. And when you compare with what it would be with the equilibrium regulation curve, you see that indeed you cannot regulate. The regulation would occur at too low concentration. So, conclusion. K90C provided us with rich thermodynamic and kinetic information. It allowed us to make a quantitative modeling of kinetic riboswitch regulation. And the interesting thing is that the results account for the correct regulation at the correct concentration, and it allowed us to make an interesting link between the k-off and the RNA polymerase posing time. So all this software now we hope are being introduced for the K90C thanks to Affinimeter. And thank you for your attention.
There is any question?
This is a question for one of the participants of the chat. He is Stefan Geschwinder from AstraZeneca, and he wants to know the first one is, I would be interested to know the limits of K-on and K-off determination using this method. This is the first one, and the second one is, can one determine long residence times, hours, with better accuracy than SPR when you have, for example, a drifting baseline?
So I understand about the first question. I understand that generally this is a very good question. Actually, I did not have time. What is the limitation of kinetic ATC? I think this is the meaning of this question. I guess. Okay, of course we cannot get kinetic information if the things are going too fast. And this is it's quite easy to see that by eye. If your injection curve do not remain very short, then you don't have kinetic information. In fact, you are limited by the response time of the instrument. Therefore, in that case, of course, you cannot get K-on and K-off. This is essentially the major limitation. The second limitation, not a limitation, but it is a very important thing to have in mind. Kinetic ITC is a model-dependent method. That means you have to propose a kinetic model. If your kinetic model is wrong, then your result will be wrong. That's very clear. So this is the second limitation. This is why, for these RoboSwitch studies, we spend a lot of time in proving that the kinetic mechanism was this one. Okay? And so, of course, if the kinetic mechanism is simply K on, K off, simple kinetic step, then, and if the reaction is not too fast, then you can get very quickly first information on K on and K off, even without doing the fit of the injection curves. We can get a rough idea of K on, K off in 2 minutes from the result of the ITC experiment. But of course, I have no time to develop that. So about the second question.
Can one determine long residence times, hours, with better accuracy than SPR?
The residence time. So I understand that it is a can one determine the residence time, right? So I understand that it. Long so low K-off. I understand that it is a question about. You agree with me? Yeah. Yeah. Yes, it is. Hours. Okay. So the real question is, can we determine very low kinetic? I mean, okay, very low kinetic. So in that case, because we are measuring a heat power. If the reaction is very low, the heat power will be very low. Therefore, we are limited by that, of course. So everything. But if you have the signal, a descent signal, if it is a long, if the amplitude of the signal is correct in ITC, and if it is very, very slow, then it is very good. It is excellent. Yeah, Eric.
Just one comment. If you are facing very fast K on or very slow K off, you can play with temperature to influence the kinetic and the concentration.
Yeah. This is. But this is the other way around. If the things are going too fast, then that means that the heat power is elevated. Then you can have an opportunity of decreasing the concentration, and then you will decrease the rate of the reaction. Of course, you will decrease also the heat signal, the heat power, but you will increase you will decrease the speed of the reaction, and this is also a possibility. So I hope I understood correctly the question. I answered correctly this question.
He asks, is this method adequate in cases of a fast reaction, as for example, intercalation of a drug between DNA base pairs?
I'm not in mind. I should, but I'm not in mind the K-on of intercalation of. Well, it should depend on the drug, but there is in the paper, in the JACS paper, a first analysis about the possibility of using KINE-ITC. So I tell the people that to look at the paper and to look at the supplementary information part, where it is also discussed in the main part. There is an equation related with the product of K-on, K-off, etc., which gives a quantitative criterion about the possibility of using KINE-ITC. But I would say this was the first answer to this question, and it was said in the paper that we have to wait for more using by different users to answer definitely about the exact possibility of using KINE-ITC. So I encourage people to try using KINE-ITC with a binding of drug into DNA. It would be very interesting.
I have another question here from another participant.
Yeah. The last question?
The last question. Yeah, this is Andrea Taladriz from CSIC Madrid. She asks, is it possible to analyze the kinetics of an aggregation process, apparently very slow, about an hour between injections?
There is no counter-indication. You just have to have a kinetic model. If you have, then you can do it.
I'm also interested in kinetics in ITC, but with interaction with membranes. With membranes, lipids, yeah. And I did some tests in the microcal equipments, And according to my tests, in the ITC 200, we cannot have very slow reactions. Somehow the signal is brought to baseline if the time dependence is very, very slow. Do you have any information on that? Do you have?
This is, yeah. If you have a very slow reaction, that means you have a very low heat power.
My impression was that the equipment, the mechanism of feedback inside the equipment were bringing the signal to zero, although it was not zero, because in the VPITC it was not zero. I had a signal, a difference between the baseline and the signal, so I had heat. But in the other one, it was brought to zero apparently artificially. So I don't know about the equipment, so I don't know if it is an artifact.
VPITC is 1 milliliter a cube, right? This is correct? So that means you have more material, more heat. Yeah, but even though. Okay? No, it's not the reason. So I have no answer to that. So we will have to have a look and... Yeah, the mechanism of feedback of the... that's a possibility. This is something, a question that we asked to Natalia is here. We have asked several times a question about exact feedback mechanism. This turned out not to be easy. I mean,
let's say that still comes down to your answer that we are.
Wait.
Okay, so yeah, I work for Microcal Marburg nowadays, and to your question, I think it's still somewhere where Philippe was alluding to that ITC 200 has smaller volume, and it has smaller specific sensitivity as to compared to VPITC. So then also there is something to your previous questions and discussions with Stefan Geschwind that related to the baseline. If we only had that absolute baseline always defined and somewhere there like a red line underlying all our experiments, we would be safe with any kind of measurements of slow binding kinetics and so on. But we don't have it.
Yeah.
So in your ITC 200 setup, signal is too small, and it is lost in the baseline because you don't also know where the baseline is. It's lost in the drift of the baseline, I would say.
It's related to the previous talk.
Yes. And it's not really exactly something fiddling with their feedback mode. It is just sensitivity. We can look at your data in between the sessions.
There is no need for more discussion.
Yes. In fact, we have discussed that with Microcal several times, and I am personally convinced that it is a feedback problem and not a problem. You are absolutely correct that the ITC 200 has a lower sensitivity per volume, but I think the problem is in any of those, and you see that in both instruments, but you see that more clearly on the VPA, on the ITC 200. If the signal is too slow, the feedback sends it too fast to baseline. In fact, you have overshooting. And so I think that's why you cannot deal. But this is, details I think we should discuss later, I guess.
By looking at the data, because you have to compare them the absolute signal, have you collected it all within the integration range for the two instruments? So that is maybe something to look at first.
If I may conclude on this point, this is a proof that we need more link with manufacturer for having the best software for KEEN ITC, because we need to know exactly how regulation is made. Yes.
Okay. Thank you again for your talk.